Morning. It's 9 o'clock here in San Diego, so we will start the webinar. Thank you very much for joining us. Today's webinar is on ligand design using the ICM 3D fully interactive ligand editor. My name is Andrew Ori, so if you have any questions after the webinar, please feel free to email me or call. During the webinar, please use the text messaging panel or the chat to ask any questions, and I'll try to address them either during the webinar or at the end. You can also follow us on social media sites, and every participant in the webinar can have a 14-day license for this software, and I will send you instructions for that after the webinar via email, along with a recording of the webinar. So today's topic is the ligand editor. So the ligand editor is widely used throughout the pharmaceutical and biotech industry for lead design and optimization. So if you have a lead compound or a reasonable binder in a crystal structure, for example, you can then modify it and try and improve the properties and the binding of that ligand. The tool was built in close collaboration with medicinal chemists at Novartis, which was extremely helpful for us to make it more user-friendly, and it also sort of rewards your chemical intuition as you start designing as well. It is a fully interactive tool whereby you can make changes to a ligand, for example, in 3D in the structure here, or in the 2D environment, and then commit this change to the 3D environment and see the effect on the score for that ligand. You immediately see the effect of the modification on predicted binding affinity via a score. So some of this interaction between MolSoft and Novartis was published in a recent paper in an ACS journal, and it's development of a global communication and modeling platform for applied computational medicinal chemists and chemists. So today's webinar topics, we're going to look at how you can read in a structure and a ligand and visualize key interactions between the two. We're going to edit the ligand in 2D and in 3D. We're going to evaluate the effect of those modifications on the score or binding, predicted binding. Show you the undo and redo capabilities, and so when you make a change, you can automatically redo it, undo or redo, and then you can save the change that you like to a chemical spreadsheet to go back to later on. Once you made a change, you can dock the ligand again or minimize it or refine it. You can add distance restraints and tethers during docking as well, so you can allow certain atoms to remain in the same place during docking if you have key interactions that you want to keep. You can also select a variety of different substituents and screen them at each point around the ligand in the environment. You can perform covalent and fragment docking and fragment linking as well. We'll show that. We'll show also methods to incorporate induced fit, for example, multiple receptor conformations, MRC, as well as allowing certain residues to be flexible during docking. And you can also design ligands purely based on the chemistry using 3D pharmacophores. So if you don't have a crystal structure but you have a number of lead compounds, you can superimpose those compounds and then modify a lead compound in the 3D pharmacophore. So for our example today, we are going to concentrate on riscovatine, which is a compound that is in phase 1 trials at the moment by a Scottish company. It's an experimental drug candidate in the family of CDK, cyclin-dependent kinase. It preferentially inhibits multiple enzyme targets, including CDK2, 7, and 9. It seems to alter the growth phase or state within the cell cycle of treated cells, and it's in clinical trials for treatment of lung cancer, Cushing's disease, leukemia, and other diseases as well. So, and there is a crystal structure with riscovatine bound to this kinase. So that is how we start our demonstration. So we need to load display ICM. So you should have an icon ICM here. And the crystal structure PDB code is 1UNL. So you go to the search tab, type 1 UNL, and we can see the two main molecules, A and B. There's, if we just go to the display tab, click and hold on ribbon, say color by molecule, it just makes it easier to see we have. The A subunit has riscovatine bound. You can see it here. This is the ligand here. So if we just double click on this in the center, that will take us to our ligand in the pocket. Okay. So all the 3D ligand editor tools that we're going to show today are in this Lig Edit tab. So you click on the tab, and basically you can work your way along these buttons. So to start with, we need to tell ICM which molecule that we have loaded. You could have many molecules here, but we only have one PDB, but you might have more. You have to tell ICM which is the ligand. So set up ligand, click here. And because there's only one small molecule, it automatically finds it, but you might need to use the drop-down button to locate it. And you can ask ICM to auto-assign formal charges and based on a pH, or you can just leave it. If you've already assigned them, you can leave it and uncheck here. You can also optimize and model peptides as well. So if you had a peptide bound, ICM could recognize it as a chemical and you can start modifying it in the 3D ligand editor. So you just go OK, and that sets up the ligand. So now we have the original PDB file here, and it's copied the ligand to a separate object called 1UNL_lig. The next button along is the setup receptor. You click on that, and you see two tabs at the top, one for protein and one for APF. APF we covered a couple of weeks ago in the webinar, which stands for atomic property fields. So this is the 3D pharmacophore option. But we're going to start off with the protein. We've got a crystal structure, reasonable resolution, so we can use that for docking and optimizing this ligand. It asks us which is the receptor object. We only have one main object here, one protein object. It's 1UNL, so it automatically finds it. You can choose to keep tight waters, which maybe have three hydrogen bonds or more with the ligand and the receptor. Or you can choose to keep all waters, or you can delete all waters. That's a choice you may need to investigate, play around with, have a look at the waters and see which ones you want to keep and not. So it's probably best to keep the tight ones to start with. And then there are three more options. So what we read in was an X-ray structure, so there are certain atoms missing from the crystal structure. There might be, if it's poorer resolution, you might be missing some atoms. And so you can choose to mask those, and so ICM won't build them, it'll just add a density around that region, or you can ask ICM to just build those missing atoms if you uncheck here. And you can you need to optimize the hydrogens and the asparaginins and glutamines for the optimal hydrogen bonding network and optimize existing hydrogens. So if you have a pre-prepared structure from another software, you could just uncheck all these and just leave it as is. Then the next step is to tell ICM where the ligand is. In some cases, the ligand, in most cases actually with the ligand editor, you're editing a ligand that's bound to the receptor. So you know where the pocket is. So you can just say make box around existing ligand, which is what I would do. But you can run the ICM pocket finder, which will find binding pockets around the protein, and then you just select the pocket. But in this example, we're going to make box around existing ligand, and you can see here we have the purple box, and this is where, so by default we're not going to be, we'll be building energy maps inside this box in order to dock. So there'll be maps for electrostatics, hydrogen bonding, van der Waals interactions, and other maps. So the idea of the ligand editor is obviously to build. So you need to make sure that if you have a particularly small starting ligand, you may want to increase the size of your box to encompass a larger portion of the pocket. generally it's usually okay, but it's best just to keep an eye on that. For example, here we have the tip of the, this is a kinase, so this is the glycine-rich loop coming down here on the N-terminal. You can see it's sort of poking out the box. You may want to increase it a little bit here. And maybe look out for the DFG here as well. But for our example, this is fine. And then you press OK, and it will generate the maps. It takes a few minutes. So I have a pre-prepared example for that. So for before we can start editing, it's best to sort of investigate the pocket. So there's different ways of visualizing how you can build, or you know, when you're building, maybe you want to flash regions where you've got clashes, for example. So you can display the receptor surface colored by binding property, which is shown here. You can display the ligand pocket surface, which is kind of the opposite of this, but it was suggested to us by the chemist at Novartis that it actually gives a better representation of where there's cavities to build into. So I will show that pocket. This is the opposite of what we see here. You can display hydrogen bonds. These we call energy circles, and they highlight clashes. So if you make a modification and it clashes into the protein, you can easily see the problems with these red clashes. These green mean that these atoms are fine, but you need to get rid of this. Stop modifying there, for example. An important thing to keep an eye on is the relaxed ligand. So you can display the original ligand that you're docking or you're modifying compared to the energetically favorable conformation. So it is possible to sort of design artifacts. So you need to always keep an eye on the strain and the relaxed ligand. You can also display unsatisfied hydrogen bonds, which might give you some regions to go and build towards. So we were at the point that we've set up everything and we're ready to edit. So this is what it looks like after you generated the maps. We see we have all the maps here. You can display them, but we don't need to do that for this example. So we're just going to look at the visualization tools at the moment. So all the visualization tools are on this side. These are the setup. Visualization is here. So we can toggle the hydrogen bonds on and off. You can see we have these two hydrogen bonds with the hinge region of this kinase. If we display in ribbon, it might make it easier for us to see where we are. Yeah. So we have the glycine-rich loop here in the N-terminal. We have the C-terminal, the DFG here. This is the hinge coming down here. The alpha C is kind of out of the way at the moment. So we can display the receptor pocket, which I showed before. So you click and hold on this button here, receptor pocket, and so we have hydrogen bond donors in blue, hydrophobic regions in green, lipophilic white, and red hydrogen bond acceptors on the surface of the protein. The opposite of that is known as the ligand pocket surface. So click and hold here and say ligand pocket surface. You can see this generates a different view, the opposite actually of the other one. So it also, you can see that we have a cavity here that we can maybe build into. And it sort of opens up, it's an easier way to see. And it also guides you, so you need to put a hydrogen bond acceptor here to make interaction with this hydrogen bond donor, for example, or matching hydrophobic with hydrophobic regions. So it's a guide. And if you click and hold here, we can undisplay that. So other things to show are, for example, the unsatisfied hydrogen bonds, and we have one on the protein here towards the gatekeeper at the back of the pocket. You can see, so you might want to build towards that, but we already have two hydrogen bonds to the hinge, which is quite normal for a kinase inhibitor. And also we might not have much room there as well. You can just double check that on the with that ligand pocket surface. You see that, yeah, There's no, there'll be, if you start trying to build there, you'd have a clash. There's no cavity, there's no volume. So we can also display the relaxed version of the ligand. So display relaxed ligand just generates another object called ligstrain here. And so when you're designing, we haven't started designing yet, but you can check how your designed ligand looks compared to the relaxed version of that ligand. You can toggle it on and off here. And when you do that, it displays these energy, these are the torsion angles, whether they're strained or not. Green is good, so all of them are fine. You can toggle these, they're called variable here. You can toggle those on and off with this button here. But if they were red, it means you've certainly added some strain to your ligand. And it would also tell you here at the top here that you have a high ligand strain. Okay, and so before we carry on, before we start designing, you may also want to look at the contacts. So if you click on this Analyze Contacts button, it will tell you what residues are in direct contact with your ligand. This X-stick zoom scale is designed so residues that are making big contacts with your ligand will be thicker in size. The sticks will be thicker. So we go OK. And we can see, for example, this leucine 133 is making quite a lot of contact with the ligand. The representation is very thick. And so we go to the table and we look down here. Oh, leucine, it's not the most, but isoleucine 10. If you double click, this one is actually making a lot of contact with the highest contact with the ligand. It's coming off the glycine-rich loop at the top here. And you can see the contact area, exposed area, and the closest distance to the ligand as well in angstroms. So this is a fully interactive table, so you can just double click on one and look at the interaction. Okay. Another way to look at the interactions is in a 2D diagram. So you click this button here, will generate the 2D diagram. Single click. There's different parameters that you can change. For example, hydrogen bond strength. If you want it to display weaker, you could lower this threshold. You can use hydrophobic distance as a cutoff for the hydrophobic interaction. You just go OK, and this will generate a 2D diagram in a chemical table here. So you can see that this is the hinge. This is making a hydrogen bond with this cysteine on the hinge region here. There's also a hydrogen bond going to the glutamine and asparagine here, and the others are coloured. We'll cover this actually more detail next week. Next week when because it's a brand new feature, and next week we have a webinar on new features in ICM. So other important things to look at are you can center on your ligand by clicking the center button. You can also, which is useful, is to change the hydrogen display. So you can say all hydrogens, or ligand all hydrogens, and the receptor hydrogens as well, just the ones that are making contact. And when you start modifying, you need to click on a hydrogen so to add a group, so it's important to display the hydrogens as well. These are the energy circles, which I'll show in a minute when we start modifying the display. Okay, so we're just going to close this down. Just had a question about the editor. This is a brand new 2D diagram map. We use everything in ICM is written at MolSoft, so this is MolSoft code. It's not using other tools. There's some other questions I might have to get back to you after the webinar on that one. Another one. Okay, so let's carry on. So now we're going to move into the ligand editing. So one way to edit the ligand is in 3D. So I'll show you the panel. You can click on a group, for example, and try that group immediately on a particular residue, on a particular atom. You can change atom types and bond types and some other more advanced things for docking, such as distance restraints and tethers. And there's full undo and redo capabilities. You can also screen multiple substituents, so you can select like 6 or 7 of these or more if you wanted to, and then just click on an atom and it will run. After developing in 3D, we realized that chemists actually quite like 2D, so you can also do it in 2D. So you have the 3D environment on the right hand side and the 2D panel on the left, and you can make changes in 2D and then immediately commit them to 3D and see the effect of the score on the score. And the editor, as we saw last week in the chemistry, ICM chemistry, it's pretty feature rich. It has a chemical property monitor, inbuilt templates, and supports SDF, MOL, INCHI, SMARS keys. Yeah, so you can. And the important thing is saving your modifications. So, as I mentioned, everything is saved in the memory, so you can undo, redo, but you can immediately add something that you like to a chemical spreadsheet and tag it as a particular series, and I'll show that. And so you can save your modifications in PDB format, chemical spreadsheets, SDF, export to Excel or HTML. And you can also save in ICM binary format, which allows you to display in the free ICM browser or on inactive ICM on the web, or you can share using iMolView on the iPhone or iPad. So if you're a computational chemist and you want to share your designs with the chemist in the lab, you can just save your environment, and then email that to them, and they'll be able to open it in the free browser or on their iPad. Okay, so I'm just going to get a new ICM session. We're using the same example with riscobutene and CDK kinase, so it's the same file. Okay, so we can start editing this ligand. You may want to change the view. We have inbuilt in the view something called Pretty View, but you can modify this any way you like. I don't particularly like the CPK purple, but in the preferences you can change that view. So here we have the ligand. We can see the hydrogen bond here, colored strong, thick spheres are strong hydrogen bond, and green is good. Weaker ones are in red. So we can start modifying, and we're going to start modifying initially in 3D. So let's look at the ligand pocket surface again. Okay. So as I mentioned, we got some space here to modify, here as well. And just to show you the energy circle, so to make a modification, we set up the ligand, we set up the receptor. The next button along is the Edit Tools button. So just for clarity, I'm going to undisplay that pocket. So you can see there's not much space here. So if we tried to add a carbon here, for example, you can see there's not much space. And so if we add another carbon, we are definitely going to have a clash. You can see you've only got room really for, well, basically there's not really room for any addition here. You see if this this region is not good to modify. We knew that anyway by looking at the surface. So unless of course there might be some flexibility in the receptor, which we'll cover later on. So that's also to be careful about. But in the current crystal structure, we have a clash at this point with this phenylalanine. So this is not a good modification that we just made. I was going to color the ligand in green, just to, so if you double click on the ligand, go to the display tab, click and hold on the stick and say color C only, which is color carbons only. I was going to color it green so you can see it a bit better. Okay, so that wasn't a very good modification. You can see the score is very poor, the strain is high, So I'm just going to undo that modification. I can go back, undo, back to the original ligand. Okay. Let's have a look. If we remember, there is some space down here on this ring. So you can see there's like a hydrophobic region we could maybe try and build into. It's going to undisplay surface. So let's see how many carbons we can fit in here without causing a clash. So you can add one, I think, and then add the second one. So after you add one, we can look at the energy circles and try second one. You see, we've only got room for one carbon. So let's just undo that one carbon on this ring, and we can look at the score. Actually, that carbon, because we were building into a hydrophobic region, actually improves the score. And so if we like this modification, we can add it to a chemical spreadsheet. So this button here says add current ligand to table. If you single click, it will just immediately add it to a table, but if you click and hold, you can say tag and save ligand. So I'm going to tag it. I'm going to give it a tag and say phenyl mod 1. You see, now we have the chemical spreadsheet here. And so if we even if we delete the ligand, we can just double click on this table and it will immediately put it back into the pocket. So we have all the information. We have the tag that I just gave it, the confirmation, the receptor that it's bound to, which is 1UNL rec 1, the score, the VLS score. You could use the lower the VLS score, the better the predicted interactions are. And then there's also some other properties. You can also add your own properties. If you were at the webinar last week or 2 weeks ago, you can do calculate properties and you can add your own properties to this table. But this is just a sort of a you know, to keep a record of your modifications. But it's also in the memory as well, so you can always go back. So okay, so that's one modification that we like. Maybe we could try in the editor panel. We have other options. So you could say maybe we could try a halogen there. So if you choose bromine and click on this carbon, it's converted to bromine. Evaluate the score, and the score has got lower. So that's a good modification. We can add it to the spreadsheet. We also have to be careful. We've added a little bit of strain, so we should really minimize that ligand or dock it, and I'll show that in a bit. So now we have two modifications that we like. We can use the editor to change bond types. So you can use, for example, double bond. You click on a bond, and you can use the eraser to erase atoms. You can use the elements here to change them, and then evaluate the score. So that actually the score went higher, so that's probably not a very good modification, probably not one you might not want to pursue. Okay, so as I mentioned, you can also edit in 2D. So we click on the 2D editor button, which is this one. This opens the 2D editor panel, and we have our ligand displayed. We also have this property panel, which you can toggle on and off. So you can play around with, actually modify it automatically on the fly, calculates the predicted solubility, log P, for example, and other properties. So maybe we could try on here something else. We could try maybe like a sulfonamide. So if you add, you can use the keyboard for the modifications, and for the atoms, or you can just click here. So if we want to try that, we apply changes to 3D, and it automatically adds that ligand, that modification here. So if we like that change, then we can add it to the table again. So I'm going to go back to the original ligand. So I can undo everything and we're back to the original one. But she's still got the bromine. We can remove that bromine and remove the carbon. Now we're back to the original ligand. So here we have a lot of other tools you can use. You can obviously define stereochemistry if you right-click on an atom, on a bond, sorry, and choose stereo. You can change the stereo. And there are other templates here, groups that you can use for sketching. So as you can see, there's tabs here as well. So the workspace panel, let's go no for that. We can edit. These are the edit tools, and this is the 2D editor. So you can toggle through these here. So we go back to the editor. So we tried interactively. We tried a few one halogen, and it seemed to improve. So we can actually try fluorine, chlorine, bromine, iodine, all in one go, along with some other groups that we can check. So there's sulfonyl, sulfonamide. So you can check as many as you like, and then we can do a virtual screen on the fly to a particular point. So this point seems like a good starting point. So I selected them, and then you just click on the hydrogen, and it would do. See in the bottom right hand corner, it's trying the groups. It's virtually screening them on the fly. And then we get a table of them ranked called modifiers tab. These are the ligands that we saved. This is a new table that we just generated, and the original ligand actually is in 8th place in terms of the VLS score, and these other groups, the bromine one is actually the best we could find, but these other groups have good, decent VLS scores as well, and the strain is good. So these are. So if you wanted to see the one with this sulfonamide, which we've already done, but you can just double click and it loads it. You could add it to the table, and you see it. Now we have it here. We can go back to the original one by double clicking here, or these are the screening results, so you can go through. Now if you want to check what you've done, you can click and hold on here and use the modification history, and this tells you, so this starts off with so we've now got 3 tables. Ligands is our record of changes. Modifiers tab was that virtual screening that we did, and the modification history tells us what we changed. So we changed an atom, we attached the chemical group, and we loaded a confirmation from the modifiers tab for this, so we can display at each point what we did. So you've always got a way of knowing how you got to a particular design. So these options are all in this button here. So these are for editing. So as we move along, we see two more options. So you can minimize the ligand, which is sometimes a good idea to do. It does a small minimization after you do a modification, but it's sometimes a good idea to minimize again, or just to check the confirmation. Or the next button along is a full docking. Okay. So just one slide from. So the docking uses MolSoft's ICM docking technology, which we'll probably talk about more next week, or sorry, 2 weeks time when we have a webinar on virtual ligand screening. So you can dock a modified ligand, or you can dock a collection of ligands like a virtual screening from a chemical spreadsheet. You can screen a database of substituents either interactively by clicking on them, or you can go to the advanced button and dock in one go a selection of substituents. You can perform induced fit docking. If you're fortunate to have many crystal structures, then you can use the information from all those crystal structures and the flexibility in the pocket that they define in your docking. So you automatically dock to an ensemble of crystal structures, which we call 4D docking. You can do receptor side chain refinement. As I mentioned, you can dock to 3D pharmacophores, covalent docking, fragment docking, so you can dock a small fragment or a couple of fragments and then link them using a database of linkers. And you can also dock using tethers and distance restraints. Okay, so for docking, let's start with the distance restraints. So if we're making this, if we're trying to design an ATP inhibitor to the ATP binding pocket, most likely these two hydrogen bonds need to be kept. So you can make your modifications and redock using distance restraints, or tethers in this case. So we click on this button here to load this panel, click on tethers, and then you can click on the atoms that you want to keep tethered. So we want to keep this atom in place. You can see now there's a red cross, and now we have an additional table here called dock restraints, and it's telling us that this atom is currently set for as a tether, and then we can, it's going to undisplay that thing. Then we can click on the atom here and choose these two to keep in place. So it's telling us that if we dock now, we're going to use these two restraints. You can toggle the display of them off. It will still use them if it's checked here. And it's telling us which atom. So it's this atom here on the ligand. And you can play around with weights, but generally this is fine. If you don't want to use it anymore, you can just check here, or you can just delete a restraint, or you can just add it back. So we have two restraints. And I'm reluctant to do the docking because it takes a little bit of time, especially with the graphics displayed, because it's about a couple of minutes, or maybe less, but depending on the ligand. But so you would redock just by clicking here. You'd get reevaluate the score, and you go from there. And it's the same with distance restraints. So if you want to add some distance restraints, you click on the button above that, which is the D. Click here, two atoms, and you can see that you can change the maximum distance, maybe increase that to 3 or something, give it a bit more leeway. And we can add another distance restraint here as well. Because we're using distance restraint, I'm going to delete the position restraint, which is the tether. So now we have two. So we have two restraints that you can play around with maximum, minimum distance, and the weight of those. Then when you redock, this area will not move. I showed that we can also dock the substituents on the fly by selecting here, but you can also add your own substituents. You can add new. So just for example, just close it, save changes, go yes, and you have that substituent added here. But we can screen all of these plus other substituents. Also, you also need to have it's going to edit it. You need to have an attachment point. It will actually automatically generate it in this case, but if you right click and choose attachment point, that will add where the substituent will be attached. This is great interactively, but there's an option to do it on the fly in the background. So you would say find group, and you just check you select the atom you want to try it on, so for example here, and then you can either calculate the full binding score, which takes a little bit longer, or you can calculate just the score for the substituent only. This would be the whole ligand, or just the substituent. You can use the system modifier list, which comes with ICM, which is a collection of these plus other ones, or you can use your own external table. If you click here, then you can use that table, and that will screen these substituents to that point on the fly. Another option is multiple position scan. So we click here, we can choose a group or more than one group, and it will try to add that group to all hydrogen atoms on the ligand, or just selected ones if you make a selection. So for you can select more than one, obviously, and do that. And you can also do a heteroscan as well, all ligand carbons. You can try it with nitrogens, oxygens, and sulfurs. And it will score those and give you a table similar to the modifiers table below. So you'll have a VLS score and other things here. So there's two different ways of doing this on the fly rather than interactively. This will do it in the background. You can also dock a chemical table. So we made this table called ligands, which was our favorite designs. You can just dock that on the fly. You go to advanced, dock table. You may have 100 compounds, so it's a convenient way of docking them. And it will redock them, assess the VLS score. And so you go to get this panel, you go to advanced. You say dock table. The input table is ligands. The effort is the length of the docking simulation. 2 is probably reasonable, but if these are particularly large chemicals, you may want to increase it to 5. Or if you've got a lot of time, you can maybe increase it to 10, just to be as thorough as possible. And the number of poses to keep, it will give you a stack of 3, for example here. The top 3 energetically favorable poses will be displayed. You can also use covalent reactions for covalent docking as well. Okay. Just want to actually go to start with the induced fit. So this was, as I've described at the moment, is all being in just one receptor. But you may want to incorporate some kind of flexibility. So probably the most efficient way of doing that is using 4D docking, and it uses an ensemble of conformations. So this is 4D docking. This is described in a paper by Ruben Abagayan's lab at UCSD and scientists at MolSoft as well. So basically you incorporate the maps from a variety of ensembles of crystal structures and then dock on the fly to that ensemble. It's more efficient than doing it individually to each one, which is the regular way of doing it. There's an additional option to allow, so by default we have this map, but you can allow certain atoms or residues to be flexible, to be explicit so they're not maps, they'll be explicit atoms. So here we have some hydroxyls that we're allowing to be flexible. So you can select certain residues to be fully flexible when docking. And there's also an explicit receptor refinement which can be used for just sort of relaxing the pocket a little bit to remove any clashes. But with these two options we need to be very careful not to select too many residues. For example, you may see artifacts being formed. So in an ideal situation you'd have many multiple receptor conformations, or you could model certain conformations where you think there's flexibility, or if you have certain residues which you know are flexible, you can allow them to be explicit. So to do that in ICM, for the side chain refinement, you can just click here and it asks you all side chains, or you can make a selection. And that runs, you can see, it's just minimizing the side chains around the ligand. This is and we'll probably see the score improve in a little bit. Okay, it takes a little bit of time. So you can see the progress here. So I'm just going to stop that. Let's just see. Yeah, I'm going to just cancel it. And so you can also, as I mentioned, you can define flexible receptor groups. So if you click on here, you can select, for example, we can select this lysine. This is the residue selection button on the right hand side. Just single click on that and we have green crosses. Just want one. So we have one residue selected. Do full side chains. Just go OK. Then when we dock, go to docking. This will be flexible. So the map, it's remaking the maps, allowing this lysine to be flexible. Okay. So the most efficient way is to, we'll just let that run for a little bit. We can have an example for the 4D docking. So open. So in this example, I have I've got some more questions I'm going to have to answer, I think, at the end. I'll have to get back to you. So we have 3 nuclear receptors, which actually it's the same nuclear receptor, but the pocket in the nuclear receptors are notoriously, they can be quite flexible. So we can see that in these 3 crystal structures of the same protein, we have different pockets in size. So we can modify our ligand and in this using information from each of these 3. So to do that, this is called 4D docking. We can select, we go to the Ligand Editor. We have a nuclear receptor inhibitor here, which we're going to edit. So I'm going to go to the Ligand Editor, go to Ligand Setup again, and choose the ligand, which is ACO1. Just go OK. And then we need to, in order to dock to this ensemble, we need to superimpose them. So we select them using, you can right click and drag over them, or you can double click in the ICM workspace, go to display, and there's superimpose button. They ask you which object you want to be static, and it's 1P8D is the one I want to be static, and just go OK. And just want to be careful it's selected. Okay, just go OK. Yeah, I didn't like the ligand being selected, but anyway, it's fine. So you can see now that the receptors are superimposed, and the pockets are, you can see the difference in the pockets. We can select neighbors of the ligand and look at the 5 angstroms, all objects, and we can just see there's like significant changes here. So it's gonna, so I made a selection and it didn't propagate to all residues, so we can use the R, and now we can see all the atoms of every selected side chain. There's some rigid parts of the pocket, but there's a lot of flexibility. So to dock to these multiple conformations, we need to select the conformations that we want to use. Go to Ligand Edit. We've already set up the ligand. Now we set up the receptor. Click here. So now the receptor object, we have 3. So we have 3 objects selected, which is defined here. We have the same questions as before, and we can use make box around existing ligand. And now it will make the 4D maps. Okay. So we get a new panel here, so we can, it enables us to, we can look at the score of this current ligand. in each one, so you can toggle through. See, in the second confirmation, the VLS score is positive, so if we look at the energy circles, we can see that the reason for that is that there's a clash here. If we look at the third object, we can reevaluate the score. We've got massive clashes down here for this one. And for the first object, the score is pretty good, because that's where the crystal structure came from. So so now we can actually design in 3D, taking advantage of the flexibility of the pocket. So we just modify in the same way as before with the panel, and then when you can redock to the ensemble by clicking and holding on the docking button and say redock in multiple receptor conformations 4D, and that will dock to that ensemble. And then you'll get a table as before with the docking results, and it will tell you which receptor conformation is preferred for each one. Okay. And just for the final example, we talked last week, 2 weeks ago, about APF. Here, which is the atomic property field method developed by Max Totroff here at MolSoft. So it has different 3D APF components, such as hydrogen bond donors, hydrogen bond acceptors, sp2 hybridized lipophilic regions, size and charge, and electrostatics. So you can superimpose chemicals and then dock to that cloud inside the ligand editor. So dock to these properties, basically. So I'm just going to load that example. I had some more questions. I'm going to have to definitely get back to you after the workshop, after the webinar. So in this example, it's called APF, or atomic property fields. Okay, and here we have some cyclooxygenase inhibitors. They're very good inhibitors. We've superimposed them, and we can dock. This is the APF fields, which I'll show you how to generate in a minute. So we go to Lig Edit. We go to Ligand Setup, and the ligand that we want to edit in this field is here, called Ligand. So click on the button here, choose Ligand as before. That just tells ICM which is the ligand. And then for the receptor, we need to select the templates we want to use, and then go to the APF tab, and it asks us which of the templates, 3 molecules of the templates. You can also use an excluded volume. So if you don't have the crystal structure, if you have a crystal structure, you can give a sort of excluded volume around the outside of the 3D pharmacophore, so you don't start building into what might be the crystal structure. But we can just use, for this example, we'll just use the atom template. Just go OK. And then we can visualize the APF by clicking on this visualization button, and you can see them here. So we have hydrogen bond acceptors, a lipophilic region, electronegative, so you can toggle these visualizations off. So now we can edit this ligand. This is the ligand we want to edit, and we can edit it in these fields. So in a similar way, you have a ligand strain. We can do a minimization to improve the strain, fits into the fields. We can use the editor buttons here to make changes, modifications to the score, evaluate the score here. As before, the lower the score, the better. It's on a different scale. You also use the eraser button and to erase the certain groups you don't like, as before. and score, and you can obviously save it to a table as we did before using atomic property fields. So that's an overview of the ligand editor. There are a couple of options which I didn't cover, but you can dock the fragments, and you can use the option to find a linker between the two fragments using, we have an inbuilt database of linkers. You can join the two or three fragments, and that's another option. The covalent docking is a similar process. You select the residue where you want the covalent bond to be made, and then you can modify the ligand using the covalent interaction. Okay. So just want to say thank you very much for your attention, and thank you very much for your questions. I'm going to have to, as I mentioned, most of them I'm going to have to get back to you after the webinar. But if you have any more, I'll try maybe it now. But if you have any questions, please feel free to email after, and I'll send information about how to get the software in a separate email. Thank you very much.